Hi everyone. Today I'm very excited to introduce you a technology we developed in the lab trying to understand splicing regulation and potentially speed up the development of antisense oligo or ASO drug. So I would like to start from a little background about alternative splicing. As you probably know, most of the human genes have alternative exons that allow generation of multiple transcript in the protein isoform from single genes. So this is an exon, and a zooming view shows the basic idea how splicing is regulated. As you can see, the boundaries of the exon, the splice site is absolutely necessary, but in addition to that, the cell require to recognize many additional regulatory sequences, either in the exon or in the intron. These signals need to be integrated together to control precise splicing. So why this is important, and this is probably provide a very important example about spinal muscular atrophy or SMA. So this is a very deadly disease called #1 causes infant death. So this disease is caused by a loss of a gene called SMN1, and this case is actually very special because in humans there's a backup copy of the gene called SMN2. SMN2 and SMN1 are almost the same. The difference is that in SMN1, exon 7 is included, which is required to generate a functional protein. In the case of SMN2, the exon 7 is skipped. So as you would imagine, if you have a way to allow inclusion of this exon, and this will be helpful for the patient, allows them to generate functional protein from SMN2 to compensate for the loss of SMN1. So this was the development of a drug called Spinraza, which takes one is suppression of intronic splicing silencer in intron 7 downstream exon that lead to the effective inclusion of exon 7 in the generation of the functional protein. This was the first effective treatment for this deadly disease that was approved by FDA a couple of years ago. So how this works? So Spinraza is an antisense oligo drug, or called ASO. It works by binding to a specific region in the pre-mRNA to block the recognition of the sequence by particular RNA-binding protein splicing factors. In this case, it blocks the splicing silencer and allow inclusion of this exon. So as you can see, the key is to identify which region can be targeted by antisense oligos. And this is not trivial because we don't have complete understanding about splicing regulatory mechanism. Historically, actually, the most effective element was identified through very exhaustive screening called ASO walk, which is to test many individual ASOs that are tied through the entire region, including exon and flanking intronic sequences, as illustrated here in the schematics. So this approach is not high throughput. You need to test many ASOs one by one, and it's very time consuming. It's very expensive. So the question is how we can speed up the process.
Antisense oligonucleotides have been proven to be extremely effective for treatment based on splicing modulation. However, the potential of ASO therapy is limited by inadequate mapping of splicing regulatory elements, SRE, and inconsistent performance of current approaches for SRE mapping. So to tackle these challenges, our lab has invented a new technology called SpliceRUSH, which stands for splicing regulation uncovered by steric hindrance. Most of the time, RNA splicing is achieved through the interaction between splicing regulatory elements and RNA-binding proteins, RBP. So ASO therapy exploits the interaction by masking the regulatory elements to prevent RBP binding. The RNA-binding CRISPR-Cas13 complex can also modulate splicing in a similar mode of action, which can be adapted to high-throughput screening and is the foundation of SpliceRUSH. To develop the technology, we construct a fluorescent splicing reporter containing SMN mini gene. There are two versions of the splicing reporter, dual in and dual X. For dual in, only when exon 7 is included, there will be TD tomato expression. On the other hand, dual X will only express TD tomato upon exon 7 skipping. The figure here shows cells transfected with dual in reporters that have different exon 7 inclusion levels, which reflect on red fluorescence intensity. For the actual screen, the splicing reporter is integrated into HEX cells together with the Cas protein. The transgenic cells are infected by a pooled guide RNA lentivirus library that tiles the entire mini-gene. By sorting transduced cells based on RFP intensity, we are able to determine the sequence of guide RNAs enriched in cells with extreme fluorescence signals. So as a side note, the code for sequencing analysis is available on our lab's GitHub page. And I've written it specifically for our study, but it is also applicable to other experiments with a similar format. The screen result identifies 3 splicing regulatory elements. The first 2 elements are well known to the field. One is a splicing enhancer in exon 7, and the other one is the intronic splicing silencer targeted by the ASO drug Spinraza. The third element is splicing enhancer in the deep intronic region. This splicing regulator is being described for the first time in the literature. We then designed ASOs targeting the newly identified intronic splicing enhancer. As expected, exon inclusion is reduced with these ASOs, suggesting splicing regulatory elements identify SpliceRUSH can be served as the basis of ASO design. So as you can see, SpliceRUSH overcomes limitations of existing methods to map SRE and allows high-throughput systematic identification of splicing regulatory elements. The technology opens up new avenues for studying regulation and finding therapeutic ASO targets for treatment.
Moving forward, we will develop these applications, but at the same time, we'll also continue technology development. And there are several things we definitely think about in our mind. For example, do the screening in the more physiologically relevant conditions, and will also further improve the throughput and sensitivity of this assay. So we are very excited to have this very unique exon-centric approach to help us understand both fundamental mechanisms of splice regulation as well as a screening tool for potential genetic medicine. Thank you.